Welcome to the deep dive. Today we're tackling something really central in molecular biology. How do you find that one crucial insight, that needle of truth, buried in just, well, thousands of complex, messy data traces?
It's a huge challenge, especially with single molecule fluorescence, microscopy, or SMFM. It's an amazing technique, don't get me wrong.
Oh, absolutely. Revolutionary, even.
Right. It lets us watch individual molecules doing their thing in real time. You see these fleeting moments, these transient intermediates or rare behaviors, things that are completely washed out when you just average millions of molecules together, like in the old ensemble methods.
Exactly, that power to see the individual story. It's fantastic, but it creates this enormous data problem, a real bottleneck. We're talking thousands, sometimes tens of thousands of these individual time traces. And analyzing that pile of data, it's often done by hand, it's slow, it's subjective.
And you need some serious domain expertise to even begin to interpret it correctly. It's a heavy lift.
And even the standard tools researchers use can fall short, right?
Yeah, definitely. Things like fret efficiency histograms, fret being first-order resonance energy transfer, that molecular ruler measuring distances.
Right.
Well, those histograms are useful, but they can totally hide a really important step if, say, only 5% of your molecules actually do it.
So a minority state gets completely lost in the average.
Precisely. Especially in complex processes like pre-mRNA splicing or translation. There are so many steps, so many potential rare states. They just vanish in the standard plots.
Okay, so you need something better, something faster, more objective, and that's where the focus of our source material comes in. META-SiM.
Right, META-SiM. It stands for Multitask-Enabled Transferable Attention Model Neural Network. Quite a mouthful.
Yeah, it is. But the core idea is that it's a foundation model designed specifically for these single molecule traces. So our mission today is to unpack how this META-SiM thing can act as a shortcut, really, to more systematic and accelerated biological discovery.
Okay, so first off, that term foundation model is key. It's a shift in thinking from older machine learning approaches.
How so? What's the difference?
Well, traditionally, if you wanted to use ML on your SMFM data, you'd train a model for one very specific task, maybe just classifying traces. This looks dynamic, this looks static.
The one-trick pony, basically.
Exactly. But a foundation model, like the big ones driving things like ChatGPT, is different. It's trained to learn general representations of the data itself. It learns the underlying language of the data, if you will.
Okay, but how do you teach a machine the general nature of a wiggly line representing a molecule's behavior? That seems abstract.
It uses a technique that became really famous in natural language processing, actually. The attention mechanism.
Attention, like paying attention?
Sort of, yeah. Instead of just processing the time trace step by step, frame by frame, the model looks at the entire trace all at once. It breaks the trace down into little segments, like trace batches.
Okay.
And it treats these patches like tokens, almost like words in a sentence, for language models.
Ah, I see. So it's not just reading left to right. It can look at the whole trace and figure out which words or patches are most important relative to others. Like this little blip here matters more because of that big drop later on.
You got it. It weighs the significance of different parts against each other. This happens through a multi-layer attention architecture. And the really crucial output is something called the embedding vector.
The embedding vector. Okay, what's that?
Think of it as a unique, high-dimensional digital fingerprint for that single time trace. It's a compact representation, a vector of numbers that somehow captures all the important stuff about that molecule's behavior, its kinetics, the Febvre rate levels, how stable it was, even the noise level.
A fingerprint. I like that analogy. And this fingerprint is used for everything the model does.
Yes, it's shared across all the tasks the model learns. That's part of the multitask magic.
Okay, so the architecture lets it learn these complex relationships. But what about the training? That's where the knowledge comes from. You mentioned multitask pre-training. What did that involve? How much data?
It involved a lot of data. They pre-trained META-SiM using 1 million synthetic traces.
1 million? Wow. Synthetic, though, not real experimental data.
Right, synthetic.
Yeah.
And that's actually the secret sauce here. These weren't just random squiggles. They were carefully designed simulations.
How so?
They were built to mimic a huge range of possible SMFM scenarios. One color, two color experiments, different speeds of transitions, different numbers of states the molecule could be in, varying levels of signal to noise. Basically, trying to cover the whole universe of possible SMFM data.
Okay, but why synthetic? Why not just use 1 million real traces? Wouldn't that be better?
Because of one critical thing. defined ground truth.
Ground truth, meaning.
Meaning you know exactly what the right answer is.
Mm.
In a real experiment, you observe the fluorescence, but you have to infer the underlying states and the rates of switching between them. You never know for sure if your interpretation is perfect.
But with simulated data, you programmed it. You know precisely which states existed and how fast the transitions were supposed to be.
Exactly. You have the perfect labels. This massive, diverse dataset, where every trace has a known correct answer, allows the foundation model to learn the fundamental patterns and relationships in molecular dynamics much more deeply than it could from messy, uncertain experimental data alone.
That makes a lot of sense. So what specific skills did it learn during this massive pre-training? What were the tasks?
It learned all the core things you'd want to do with SMFM data all at the same time. Classifying traces, segmenting them into different parts, analyzing photo bleaching steps,
Okay.
kinetic fingerprinting, which is about measuring the actual rates of transitions, and importantly, trace idealization.
Idealization. That sounds like smoothing out the noise.
It's more than that. It's about taking that raw, noisy signal and extracting the underlying sort of ideal step-like transitions between distinct states, like different FRT levels. This gives you the kinetic parameters, the dwell times in each state. It's something often done using older methods like hidden Markov models, HMMs.
Right, HMMs. Those are statistical models that assume the molecule hops between hidden states, and the model tries to figure out what those states are. But they can be tricky to use, need expertise.
They really do. And they often struggle with very complex dynamics or subtle changes. META-SiM learns to do this idealization task, but within this broader multitask framework.
And the big benefit of this foundation model approach is efficiency, right? Because it's already learned so much.
Huge efficiency gains. Since META-SiM is pre-trained on that giant synthetic dataset, researchers don't need to start from scratch. When they want to apply it to their specific biological system, their protein, their RNA, they don't need another massive training dataset.
This is the part that sounds almost too good to be true. You mentioned minimal fine-tuning. What does minimal actually mean in practice?
It means sometimes needing as few as, say, 100 labeled examples from the user's own experiment to get high performance.
Just 100 compared to...
Compared to potentially thousands or tens of thousands you might need if you were training a deep learning model from scratch for that specific task. We're talking maybe a 10-fold, even up to a 100-fold reduction in the amount of labeled data required.
Wow. That makes customizing it for your own experiment way more practical. You could probably do that fine-tuning on a regular laptop.
Absolutely. That's the goal, making this advanced analysis accessible without needing supercomputers or huge data sets for every new project.
Okay. Practical, efficient, but how well does it actually perform? How reliable is it? They benchmarked it, right, against other methods.
They did. For instance, in that trace idealization task, extracting kinetic rates in Fairfax states, they compared META-SiM against 14 other state-of-the-art tools. It performed incredibly well, very consistent. When it predicted kinetic rate constants, like the rate of going from state 1 to state 2, K12 or back K21, the deviations from the known mean values were really small, like 3.6% and 6.8%, respectively, often outperforming many of the other specialized tools.
That's impressive consistency. What about other tasks, like that kinetic fingerprinting?
Yeah, they had a good example there, trying to detect a specific point mutation in the EGFR protein, which is important in cancer. Compared to using a conventional HMM-based analysis for that task, META-SiM increased the sensitivity catching the mutation when it was there by about 1.4-fold.
Okay, better sensitivity.
And importantly, it increased the specificity correctly identifying when the mutation wasn't there by about 3.75-fold.
Wow, much better at avoiding false positives. So it's not just faster, it seems more reliable, too.
The foundation model approach seems to provide a real leap in robustly characterizing these complex dynamics.
All right, so the model itself is powerful. It can process traces accurately and efficiently, but the ultimate goal is biological discovery, right? Finding something new. How does META-SiM help a researcher see that needle in the haystack, that rare event?
That brings us to the visualization side of things, the META-SiM Projector.
Okay, a projector. What does it project?
It projects those high-dimensional embedding vectors, the digital fingerprints we talked about, into a space we can actually see. It's a web-based tool, so you can access it in your browser.
How does it project them? Those vectors are high-dimensional, right? We live in 3D.
Exactly. It uses dimensionality reduction algorithms. You might have heard of techniques like UMAB or PCA or TSNE.
Vaguely. They squash high-dimensional data down. Right?
That's the idea.
Yeah.
Think of them as really smart mapping tools. They take that complex fingerprint vector for each trace and place it as a single point in a simple 2D or 3D scatter plot, like a cloud of points.
Okay, so every single molecule's time trace becomes one dot on my screen.
Precisely. And the key is that the way these algorithms place the dots isn't random. Traces with similar embedding vectors, meaning the molecules behave similarly, end up close together in this 2D or 3D space.
So clusters of dots mean groups of molecules that had similar dynamics, maybe similar FR8 states, similar dwell times.
Exactly. If two dots are neighbors, their traces likely share key attributes, kinetics, signal quality, FR8 values. If they're far apart, they represent distinctly different behaviors. It gives you an intuitive visual overview of your entire data set.
That sounds incredibly useful for just exploring the data. Is there a standard map?
Yes, that's where the SMFM atlas comes in.
An atlas, like
Yeah.
a world map for molecular dynamics.
Kind of. It's a standardized UMAP projection space that was built using those original 1 million simulated traces. It provides a universal coordinate system.
How does that help?
A researcher can take their new experimental data, calculate the embeddings using META-SiM, and then project those embeddings onto this predefined atlas. Instantly, they can see how their molecules behave, their Fret states, their transition rates, compared to this huge library of known simulated behaviors.
Ah, so it helps standardize things. You can compare results across different experiments, maybe even different labs, using this common reference map.
That's the goal. It helps avoid the apples to oranges comparison problem that can happen when everyone analyzes and visualizes their data slightly differently.
Okay, visualization is powerful, but META-SiM also provides quantitative metrics, right? For quality control and discovery.
Yes. Two key ones highlighted in the source material. First, for quality control, there's the label self-consistency score, or SCS.
SES, what does that measure?
It basically checks the quality of the labels that you, the user, might assign to your traces. Let's say you manually classified some traces as type A and others as type B.
Okay.
The SCS looks at a given trace you labeled type A and checks its nearest neighbors in that high-dimensional embedding space, the traces the model thinks are most similar based on their fingerprints. It asks, are most of this trace's neighbors also labeled type A?
Ah, so it's the model checking the human's work for consistency.
Pretty much. If a trace you call type A is sitting right next to a bunch of traces you call type B in the embedding space, the model flags that. Its SCS score will be low, maybe below 0.8, which is the suggested threshold.
So an SCS below 0.8 suggests, maybe I made a mistake in my labeling, or my categories aren't defined very well.
Exactly. It points to potential human errors or inconsistencies in how you're analyzing or grouping your data. It's a tool to help standardize your own manual analysis process.
That's clever. Okay, that's quality control. What about the discovery metric, the one for finding the rare stuff?
That's the Local Shannon Entropy, or LSE. This is really the key for finding novel behaviors.
Local Shannon Entropy sounds complex. What's the core idea?
Think of it simply as a rarity score. For each point, each trace in the embedding space, LSE measures how diverse the experimental conditions are in its immediate neighborhood.
Okay. What does that tell you?
Well, imagine you have data from several different experiments or conditions plotted together. If a point has high LSE, it means its neighbors come from many different conditions. That behavior is common. It shows up all over the place, probably a stable general mechanism. But if a point has low LSE, it means its neighbors almost all come from just one specific condition or a very small subset of your conditions.
Ah, so low LSE means this behavior is rare, it's specific to only certain circumstances. That's the potential discovery.
Precisely. It flags behaviors that are highly condition-specific. These are often the most interesting, the transient intermediates, the responses to a drug, the effect of a mutation, the things that differ from the baseline.
That's a really powerful concept. Let's make it concrete. The source mentions an example using a transcriptional elongation complex, dataset D4, and changing salt concentration, KCl.
Right. They were looking at how this complex behaved at different concentrations of potassium chloride. Using traditional FRET histograms, the changes were really subtle, hard to pin down.
But with LSE?
When they visualized the data using the META-SiM Projector and colored the points by their LSE score, something popped out. The low LSE points, the rare behaviors clustered completely differently at low salt, 50 mm KCl, compared to high salt, 1000 mm KCl.
So the rare events were salt-dependent.
Exactly. The LSE analysis revealed this subtle shift from a low fret state being prominent among the rare events at low salt to a high fret state being prominent among the rare events at high salt. It was a kinetic change specifically highlighted by LSE that was completely buried in the overall population average. LSE pointed right to it.
That really shows the power. Okay, this brings us to what sounds like the ultimate stress test for META-SiM, analyzing pre-mRNA splicing in yeast.
Yeah, it's a notoriously complex system, a real beast. Lots of components, dynamic rearrangements, multiple steps.
Perfect place for important intermediates to hide.
Absolutely. So they used SMFM to watch a pre-mRNA molecule as it went through, or tried to go through, splicing. They could stall the process at specific steps by, for example, leaving out essential protein cofactors like certain helicases called PRP2 or PRP22.
These helicases are like motors that drive the process forward.
Kind of, yeah. They help rearrange the splicing machinery. So by removing them, they could trap the system in certain states.
And they applied META-SiM and the LSE metric to this incredibly complex data set.
What happened? This is where it gets really impressive. The paper reports that users who had no prior specialized knowledge of the intricacies of splicing.
Okay, so not splicing experts.
Right. They were able, using META-SiM and focusing on the low LSE traces in the projector, to identify a previously unknown intermediate state in the splicing pathway.
Wow. A novel state found by non-experts just by following the LSE signal. How quickly?
They apparently did this within about a week, which is incredibly fast for discovering a new intermediate in such a complex system.
What was this intermediate state?
It was represented by a rare minority population of traces showing low FRE values. It corresponded to a more extended conformation of the pre-mRNA, and it seemed to be involved in selecting the correct spot for the final splicing cut, the 3-foot splice site.
And was this real, or just a data artifact?
It turned out to be real. Later biochemical experiments, completely independent, confirmed the existence and importance of this state. It only appeared significantly when both those PRP2 and PRP22 helicases were functional.
So META-SiM, specifically the LSE metric, filtered out the noise and highlighted this tiny, rare population that held the key discovery. Something missed by traditional analysis and even older, advanced clustering methods.
Exactly. It demonstrated how this approach could genuinely accelerate discovery by systematically isolating these rare, condition-specific behaviors.
The implications are pretty huge then. META-SiM seems to remove a major analysis bottleneck, improve objectivity, and maybe even democratize SMFM analysis.
That's certainly the potential. By making sophisticated interpretation more accessible, even to researchers who aren't deep experts in computational biophysics, it could really broaden the use of these powerful single-molecule techniques. And the projector lets you see all this, the clusters, the outliers, the LSE scores, without needing complex manual processing first.
It shifts the focus from wrestling with the analysis tools to actually thinking about the biology, generating hypotheses.
That's the hope.
Okay, that brings us to our final thought, a look towards the future. We've established that META-SiM creates these rich, informative fingerprints, the embedding vectors. What happens if you connect those to something like large language models?
Now that's a fascinating prospect. The authors touch on this. The idea is that future versions could be multimodal.
Multimodal, meaning?
Meaning they could integrate different types of information. You'd have the META-SiM model encoding the complex dynamics from the single molecule data into these embeddings.
Right, the fingerprints.
And you could potentially connect those embeddings to a large language model that's been trained on, say, the vast corpus of biological literature.
Okay, so the LLM understands biology text, and META-SiM understands the SMFM data. What could you do with that combination?
Imagine, instead of just seeing a cluster of low LSE points on the map and having to figure out what it means, you could ask the system directly, in plain English. Like, explain the potential mechanistic difference between the molecules in this main cluster and these rare low LSE molecules over here, considering these low LSE ones only appeared when I added drug X.
And the system could, what, look at the dynamics encoded in the embeddings for both groups, correlate that with the experimental condition, drug X, and maybe query its knowledge base from the literature to suggest a hypothesis.
Exactly. It could potentially reason about the data, describe the differences, maybe even propose explanations based on known pathways or molecular interactions. It could dramatically speed up the cycle from data observation to biological insight.
Merging high-resolution experimental data directly with advanced computational reasoning and language. That's quite a leap.
It really is. Moving beyond just analysis to interpretation and hypothesis generation facilitated by the AI.
So we'll leave you, the listener, with that thought to ponder for your own work. How might integrating sophisticated high-dimensional data from your field, whatever it is, with the reasoning and natural language capabilities of modern AI, how might that accelerate discovery and understanding in your area of curiosity?
It's definitely something to think about.
That's all for this deep dive. Thanks for joining us.